Thanks, Tom, for the introduction and the invitation to speak. Also, if you can hear me, thanks, David. It's always good to have fans. Let's see. So I want to make sure I stay on time. So I'm going to restart this. No. Okay, good. All right. Oh, yeah, there we go. All right. So I'm going to talk this morning about our work in the lab on substrate targeting by protein kinases. I have to apologize ahead of time that I'm going to have to bolt right after my presentation at the halftime show. I usually like to stay around to talk to people, but if there's anything of interest, please send me an email or give me a call. Okay, so I think I don't need to tell this audience that protein kinases have emerged as major targets for targeted therapy in cancer. And I think for those of us who are on the basic research side, it's sort of including myself, sort of a foregone conclusion that amongst this large and diverse group of proteins, more than 500 in humans, that there are many, many new drug targets just waiting to be discovered. Okay. However, if one takes a look at the reality of drug development and drug discovery targeted towards kinases, one sees a very different picture, and that is that most drug discovery has really been focused on a very small number of targets. And I think this was summarized best by my collaborator, Stefan Knapp, in a recent review that he published that he called the untargeted cancer kinome. And what he's done here is charted a comparison of basically how well studied a kinase is and how much effort has been put towards discovering drugs to target it. So on this graph on the left, what they've done is they've ranked all of the kinases, 1 to 500, based on the number of citations in PubMed, basically as a measure of how well studied that kinase is. So that the best studied are here at the top and the least studied, understudied at the bottom. And what they've done on the right as a gauge of pharmaceutical industry interest is mine the patent literature for the number of citations of the same kinases. So the kinases are in the same order here on the left as they are on the right. And what you can see is that there's actually a pretty good correspondence between the pharmaceutical activity towards a kinase and how well studied it is. Okay. Now maybe that's not so surprising, and you might say that that's as it should be because these kinases that we've been paying our attention to, focusing most of our attention to for the last couple of decades, are clearly the most important ones, and these are the ones that are causing cancer, and these are the ones that we want to target. But if you actually look at the data, you see a different picture. And that's also shown in this graph on the left. And what they've done is with these orange and pink bars, they've indicated those kinases that have come out of siRNA screens for proteins or genes that are important for cell proliferation and cell survival, and also kinases where you see recurrent mutations from deep sequencing efforts that might potentially mean that these are oncogenic drivers. And what you see is that there's basically no correspondence whatsoever between kinases that are presumed to be important in cancer and how well studied they are. Okay? So we have a problem here, and like I said, I mean, the optimistic view of this is that it means that a lot of these guys down here are drug targets waiting to be discovered. But how is it that we go from this situation to investigating some of these more exploratory targets, if you will? Because you don't want to just take a hit from an siRNA screen and then go right into a high-throughput campaign to develop inhibitors without some kind of target validation. So one option would be to wait about 300 more years in order to compile this kind of information for all the other kinases. But the other idea would be that we need new methods that allow one to functionally characterize kinases on more of a kinome-wide scale, to better understand them, better understand their biology, better understand their biochemistry, and understand what their substrates are. And these are all important factors that go into development of drugs to target kinases. So what I'm going to tell you about today is work in our lab and how we've carried this into more of a high-throughput platform to allow us to characterize large numbers of kinases and hopefully provide the kind of tools and functional information that might drive drug discovery. So the main interest of my group is understanding how protein kinases are targeted to their downstream substrates. And one can think about protein kinase substrate targeting as involving a number of different mechanisms. So for example, a kinase and its substrate, at least transiently, have to co-localize to the same subcellular compartment. Then one has physical interactions between kinases and substrates. So it sort of goes without saying that the catalytic domain of a kinase has to directly interact with the site of phosphorylation, the serine 3 tyrosine residue in the substrate. But as most of you probably appreciate, Kinases also recognize the surrounding sequence, so kinases are sequence-specific, and they target particular sequence motifs at sites of phosphorylation, and we'll be looking at a lot of that kind of information today. And then in addition to these sort of proximal or direct interactions, you also have what are sometimes called distal interactions or docking interactions that are between regions of the substrate separate from the phosphorylation site and other parts of the kinase, sometimes other protein domains. And one can think of the use of adapter subunits and scaffolds as being sort of an extension of this idea of docking. So one of the things that's really driving a lot of our research is the idea that really each one of these aspects of substrate targeting, whether it be localization or docking, is mediated by short linear amino acid sequences found in the substrate. So for example, a nuclear localization signal or a sequence motif that binds to an SH2 domain or something along those lines. And so one can think of the use of specific combinations of short linear motifs as specifying a code that directs particular kinases to particular substrates. And put quite simply, what my lab is looking to do is to crack these codes. And one of the simple ideas, or one of the simple reasons we would want to do that is that if you understand the sequence rules that direct kinases to their substrates, you can then go looking in protein sequence databases to find new proteins that obey those rules that might be candidate substrates of those kinases. And a lot of the lab is interested in finding new substrates for kinases, though I won't be talking about that work today. Now, okay, so I hope that you may have some idea that it's important to understand these sequence motifs and figure out what they are. How do we do them? Well, there's sort of a problem of numbers. And that's if you even think of a relatively short motif comprising 9 residues surrounding, say, a site of phosphorylation, the number of potential sequences there is 20 to the 9th power, or 500 billion. So that's a mind-bogglingly large number. It's impossible to get at those sort of numbers of sequences through individual analysis. And so in order to determine these sorts of motifs, identify these kind of sequences, my lab uses a variety of peptide and protein library screening methods. And the ones I'll be talking about today are a positional scanning peptide library method, and I'll explain what that means in a moment, that allows us to identify sequence motifs at the phosphorylation site of a kinase. What I'm also going to talk about is our adaptation of those methods to a microarray format that allows us to analyze kinases in a high throughput manner. Okay, once we have these sequence motifs, what do we do with them? Well, I mentioned that there are applications in substrate identification. We're also very interested in the structural basis of substrate specificity by kinases, and I won't be talking about that work today. There are 2 applications that I'm going to allude to today. One is the use of these kind of motifs in inhibitor discovery. So if one knows the sequences that are targeted by a kinase, you can make a consensus peptide substrate, and that's usually a very useful tool for high throughput screening. And then the thing I'm going to spend a little bit more time talking about is using these probes, using these sequences to probe the activity state of kinases in crude systems, and I'll explain more about that in a little bit. Okay, so to talk about our main method in the lab for determining sequence motifs for targeting by protein kinases, we use what's called a combinatorial library, a kind of combinatorial library called a positional scanning combinatorial library shown here. And what we have here is a set of about 180 peptides. And each of the peptides has the sequence that's shown here in the slide. Each of them, so this we're going to be talking today exclusively about tyrosine kinases. So each of these has a single tyrosine residue that's fixed in a central position that's phosphorylated by the kinase. We have a similar set of peptides where we have serine and threonine residues in the center that I won't be talking about today. So you have a tyrosine in the middle, and then surrounding that tyrosine are mostly these positions that are labeled X, and those are random mixtures, so equal molar mixtures of all the amino acids, except we leave out tyrosine because we want all the phosphorylation to happen in the middle. And then in the context of that mixture, in each peptide, we have a single residue at a single position from 5 residues upstream to 4 residues downstream that is fixed as a single amino acid. And what we basically do is scan through these 9 positions and systematically substitute each of the 20 amino acids. So the total number of peptides in the set, we have 9 positions, we have 20 residues, that makes 180 peptides. And then what we do is a simultaneous kinase assay to determine the relative rate of phosphorylation of these peptides, and that tells us which residues are preferred at what positions in the amino acid sequence. So to facilitate that assay, we have all these peptides tagged at the C-terminus with biotin. So the way it works is as follows. We're now running this routinely in a 1536 well plate format where we have the 200 peptides arrayed, just 2 microliter reactions in each well, and we use these arrays of stainless steel slotted pins for all the liquid handling steps that allow us to transfer very small volumes of liquid very precisely from stock plates to reaction plates. And then when the reaction is done, this is a radio-label kinase assay, all of the aliquot of all the peptides is spotted onto a membrane that's impregnated with avidin or streptavidin, so that captures the biotinylated peptides. We wash it extensively and then quantify radio-label incorporation. And the output that you get is an array of spots. And I realize that I'm probably the only person here who looks at arrays of spots and gets meaningful information. So I promise you, I only show this to you to give you an indication what the raw data looks like. Any real data I'm going to be showing in heat map format or in chart format.
Does your therapist show you this as well?
Yeah, that's right. Let's not go there. So anyway, so I'm just showing you an example here, which is the CSARC kinase that we recently profiled in a collaboration with Hongqing Chen's lab, University of Melbourne, and just to illustrate that there are certain spots that are darker than others, and those are the ones that are phosphorylated better, and those are the residues that are preferred by the kinase. But the other thing I want to point out is if you take a row like this one, you can see that there are some positions where you get very little phosphorylation, and these sort of negative selections, that is residues that are disallowed by the kinase, are actually just as important as the residues that are allowed in terms of defining the sequence motif of the kinase. Okay, so we have this method, and I mentioned that we're very interested in trying to do sort of large-scale analysis of many kinases. And so one of the projects that we wanted to take on was an analysis, maybe somewhat ambitious analysis, of the entire human tyrosine kinome. Okay, so instead of just doing one kinase at a time, let's look at all the tyrosine kinases. So why tyrosine kinases? Well, as I'm sure you know, most of the well-characterized oncogenic kinases are tyrosine kinases, Abl, Sark, EGF receptor, MET, etc. And most of the clinical inhibitors are targeted tyrosine kinases, with a couple of exceptions, of course. And from a standpoint of the kind of information that we might get out of these screens, in terms of substrate identification, there's been extensive mass spec-based phosphotyrosine proteomics done in cancer cells, some in tumors, some in normal tissues. That gives us a large data set of known phosphorylation sites where the kinases are not known for retrospective analysis, and this might allow us to use our data to link known tyrosine phosphorylation sites to their phosphorylating kinases. This is work that is actively underway that I'm not really going to talk about that much. But also we might be able to give us a set of probes to allow us to analyze all of the tyrosine kinases. And again, that I'll go into a little bit more detail about. Okay, so in order to do a large-scale analysis of tyrosine kinases, we knew we were going to have to do some additional methods development. Okay, and the simple reason was that the methods that we have, while they work well, are not particularly fast, and they require a fair amount of protein kinase. And if we want to do this in a really high throughput manner, we're going to have to miniaturize it and make it faster. And so I was fortunate enough to have a really talented postdoc, Yang Deng, join my lab a couple of years ago, and he was trained as a graduate student in protein microarrays. And so he basically set about to adapt our peptide library screening method to a microarray format. And this scheme here summarizes what was probably about a year of work on his part in kind of getting the method working, and it just kind of indicates how we're doing this. Basically what we're doing is we're taking glass slides, commercial glass slides that are derivatized with polyethylene glycol that is modified with biotin. So they're biotin-coated slides with a hydrophilic surface. And then what Yang does is he layers on top of that slide avidin, or rather an avidin mutant whose sole tyrosine has been removed so that it's not a potential substrate for tyrosine kinases. So avidin is a homotetramer, and so when you coat a biotin slide with avidin, some of the biotin binding sites adhere the avidin to the slide, but you have a couple of biotin binding sites available to bind to things such as our biotinylated peptides. So those are immobilized through those available sites. And so this is spotted in high density, and you get an idea of the density here, about 400 spots per 1.44 cubic square centimeters. And we can, because it's high density, we can spot multiple sets of the peptide on the slides and then partition the slides so we can analyze multiple kinases at once. And within each partition, we have the whole set of peptides spotted in duplicate. That's shown here. And this is real data with the non-receptor tyrosine kinase FES, and it just gives you an idea again of what the raw data looks like. But all the work we do is with quantified data from this output by quantifying the spot intensities. So as a pilot project, before doing the entire tyrosine kinome, we first decided to do all the non-receptor tyrosine kinases, if for no other reason than it was kind of a smaller, better well-contained set, a less ambitious project than all the tyrosine kinases. And so there are 32 of them in human. And so first we had to make kinases, and this was actually the real bottleneck, was getting all of these kinases in recombinant form. But we took advantage of gateway technology. We could get gateway entry clones for almost all of the non-receptor tyrosine kinases. Those that we couldn't get, we generated ourselves, recombined in one step into a mammalian GST fusion vector, and expressed by transient transfection in HEK293 T cells. And the great thing about doing this in a microarray format is that you need vanishingly small amounts of kinase. And so that meant that Yang was able to get sufficient kinase to basically last a lifetime in terms of doing these microarray analyses from a single well to 6-well plate. And this really enables this sort of large-scale analysis because we're not spending a lot of resources and time producing kinase. And in any case, you purify the kinases through affinity purification and then subject them to microarray screening, and this is what the data looks like. And again, I don't expect that you're going to get a lot of useful information about this. This is just a slide that basically says, look, we did it. But I will point out a couple of things. You can tell by the more intensely colored, this is a heat map where the positive selections are red and the negative selections are green, and you can tell from that where the most selected positions are, at least the -1, also the +1 and +3 positions. But rather than kind of dwell on individual points of data here, I want to talk about what we're primarily doing with the data. So one is obviously trying to identify new substrates for kinases, as I mentioned before. But the other thing that we're trying to do is develop probes that will allow us to profile kinase activity in crude systems. And so the basic problem is this, that we want to be able to define the repertoire of active kinases, the full repertoire of active kinases in whatever crude system you look at, whether it be a tumor extract or a cell line extract, or what have you. And I think this is important because if you know what kinases are active in a particular sample, these are potentially kinases that, for example, might contribute to cancer progression, and also might be important for identifying relapse mechanisms for targeted therapies. Okay, by looking at what kinases come on in the presence during therapy. Okay. Now, why do we need special probes to do this? Can't we just do expression profiling? Well, the problem with that is that, as most of you probably know, protein kinases are not constitutively active enzymes. They actually toggle between an inactive and an active state, and often phosphorylation of the kinase itself is that toggle. Okay. So the level of a kinase is really not a good indicator of the activity state of the kinase. And of course, it's the level of kinase activity, not the level of kinase protein, that determines the signaling output. So in order to address this problem, we decided to take an approach that was pioneered by Ben Cravatt at Scripps and Matt Boggio at Stanford in the development of so-called activity-based probes, or an activity-based protein profiling approach. And the way all of these probes work is that you have some sort of chemical entity, so some sort of chemical probe, that binds specifically or binds in an activation-dependent manner to the protein of interest. So in this case, a kinase in its active state. And you need to covalently tether, you need to covalently link the probe to the kinase. And in our case, we're doing this by photo cross-linking. We install a photoaffinity label. And then you have some sort of reporter group, in our case, biotin, which allows you to read out the fraction of probe bound to kinase. Okay, and you can do this by pull down with a mobilized avidin or by a mutablotting type protocol. Okay, so you may have guessed this, but our probes are coming from peptide sequences. And the reason for that is simple. Protein kinases, when they go from their active to inactive states, typically undergo conformational changes. And there's a key conformationally flexible region of the kinase that kinase people will know as the activation loop that tends to change in conformation between the active and inactive states. And it turns out that this activation loop forms part of the peptide substrate binding site. And that's illustrated here. On the left, we have the protein crystal structure of ABLE in complex with a peptide substrate, and in green you can see the peptide binding site. And here on the right, you have ABLE in complex with STI-571, imatinib, Gleevec, which induces an auto-inhibited conformation in ABLE. And what you can see is it occludes the peptide binding site. So that suggests we could use peptide probes to specifically read out the active state of kinases. And of course, we get our peptide sequences from the microarray data. So the first thing that Yang did, actually, before making any probes, was to make a set of consensus peptide substrates for each subfamily of non-receptor tyrosine kinases. And this is the consensus that comes out of the microarray data. And then to see if these were indeed good substrates for the kinases, he did what I call the grid experiment, where he took each one of these peptides and looked at the phosphorylation rate by each one of the kinases, or a representative member of each one of those kinase families. Now, if this experiment had worked perfectly, what you would be seeing is a red line that was diagonal across the center. And of course, it didn't work perfectly because some kinases will phosphorylate more than one peptide. And in a couple of cases, for whatever reason, the peptide that we designed for a particular kinase was not the best one, but in most of the cases, this actually worked pretty well. And this gives us peptide sequences for incorporation into probes that I'll show you in a moment. But the other thing that I think is nice about this is that we now have essentially a universal peptide set for assaying non-receptor tyrosine kinases. And these are useful tools for high-throughput screening, because if you want to do a high-throughput screen, you need an assay. And the better your peptide is, the more feasible that assay is, because the amounts of kinase you'll need to use, for example, are much lower. Okay, so back to the probe generation. We tested out the idea using ABLE, because we know a lot about how ABLE is regulated, and it would give us a good ways to test out whether we could really probe the active state of the kinase. So Jan starts with a consensus ABLE substrate that we call ABLEtide. Other people call it ABLEtide 2. And then he takes a position that we know is not important for selectivity based on our microarray data and replaces that residue. It happens to be at the +2 position, 2 residues downstream from the tyrosine. He replaces that with an azidophenylalanine group, so a photocross-linking group. Okay? So if we mix this probe with recombinant ABLE and irradiate, we see incorporation of biotin into ABLE, as judged in this experiment, and we can compete away labeling with increasing concentrations of the unlabeled peptide, and that indicates that the labeling is specifically happening at the active site. Okay? And we know this is at least somewhat specific for ABLE, because if we mix equal amounts of ABLE and SARC in a test tube and treat with the probe, most of the label goes into ABLE and very little of it goes into SARC. Okay, so we can label ABLE and we can label it through the active site. Can we label in an activity-dependent manner? We assess this in a couple of ways. So first of all, we know from the work of a number of groups, including Tony Koleski and Rick Van Etten, that ABLE is positively regulated by phosphorylation. So if we phosphatase treat recombinant ABLE that we purify from cells, we reduce the catalytic activity. This is wild-type ABLE shown here, and here's phosphatase-treated ABLE. The activity goes down. And we can also increase the activity of ABLE by making specific mutations. So there are intermolecular interactions that hold the activity of ABLE in check, and if we make a triple mutant that's been reported in the literature, we can turn on ABLE. Okay? And so notably, manipulations that change the activation state of ABLE, either decrease its catalytic activity or increase its catalytic activity, have a corresponding change in labeling by our peptide probe. Okay? So if we phosphatase treat, we get less labeling. If we make this constitutively active mutant, we get more labeling. Okay, so that's one way. So the second way that we showed this was by using small molecule inhibitors. And as I alluded to previously, SDI-571, Gleevec, is sort of a special inhibitor because when it binds to ABLE, it induces an inactive conformation. And a conformation that, based on crystal structure, should be inaccessible to peptide substrate binding. So based on that, one would assume that our probe should not label ABLE if it's been treated with SDI-571. However, there are other inhibitors of ABLE out there. So dasatinib, which is a dual SARC-ABLE inhibitor, is sort of a more conventional protein kinase inhibitor that binds at the ATP binding site, but actually binds preferentially to the active conformation. Okay? And so, if anything, treatment with dasatinib, not only should it not decrease labeling, it might increase labeling. And this is exactly what we see right here. So again, here's that constitutively active mutant as a control. It's labeled better than wild-type ABLE. Here's wild-type ABLE treated with SDI-571. There's wild-type ABLE treated with dasatinib. Okay, so the last piece of data that I'm going to show is an experiment where we've been trying to use these probes to detect endogenous ABLE here in K562 cell lines. This is a CML cell line that has the BCR-ABLE fusion. And I have to say that this data is somewhat preliminary. It works some of the time. It doesn't work at other times. But when it works, all the controls work, so I'm at least somewhat confident in it. And so I'll show you what I mean. So basically, if we treat K562 cell lysates with our probe, we can label ABLE in the lysate, BCR-ABLE specifically in the lysate, and that labeling is decreased by SDI-571 and increased by dasatinib, as it was up here with recombinant overexpressed kinase. Okay, so to summarize basically this part of the talk, we've identified kinase phosphorylation motifs on a large scale enabled by this miniaturized microarray peptide screening, and this has given us sort of an array of new tools for high throughput screening and a comprehensive set of substrates for kinases. And in addition, we've been able to develop peptide-based affinity labels that target specific kinases in an activity-dependent manner. And of course, since we now have this set of peptide sequences, we should be able to tune the reactivity of those probes to any kinase that we like simply by changing the amino acid sequence. Okay, So I'm going to close by thanking people in my lab, and I primarily want to thank Yang Deng. He's now left the lab. He's now postdoc with Mark Saltzman, but he did both the microarray work and the probe development work. And I should also credit Jane Liu, who did the peptide library screening that I showed previously, and other people in my lab as well. And in the way of support, I should also acknowledge Titus Bogan and Nilda Elistin Velasquez in his lab, who provided Jack Kinases for microarray screening, and Tony Koleski provided Able for those projects. And in the way of support, I do want to specifically mention a Yale Cancer Center Pilot Project Award that we got some years ago that was actually quite helpful for us in generating preliminary data for a later award that we got for the NCI to develop this microarray work. And I thank you for your attention.